Great. Thank you very much. Very happy to be here, even though remotely, to share some thoughts with you on agency. And because there are many details that go into all the things I'm going to say today in 1 hour, you can follow all the downloads, the software, the data sets, everything is here. And this is my personal blog where I write things around what I think all of these things mean in terms of a bigger picture. So what I'd like to do today is kind of three things. I'd like to introduce some of the ways I think about this field in general and some kind of broader philosophical approaches. And then I'm going to show you some very specific examples of the kind of thing I mean, because it is very important to me that these conceptual frameworks engage with the empirical science. And so I want to show you that that actually we now have ways of studying all of this, and that these kinds of ideas do lead to new discoveries, which I think is very important. And then at the end, I'll say some more, maybe some more controversial things around what I think the future is going to look like. So the first thing I want to think about is the story of the electromagnetic spectrum. So back before we had a proper theory of electromagnetism, two things were happening. One is that we had all kinds of phenomena which were thought to be different, and in particular, they used different terminology. So we had lightning, and we had static electricity, and we had magnets, and we had light, and all of these things. And people were completely convinced that these are different things. If you tried to speak of them sort of using the same terminology, people would tell you that you're committing some sort of a category error, that those are fundamentally different kinds of things. So that's the first situation. And the second was that because of our own evolutionary history, there were massive regions of the spectrum of which we were completely unaware. We could not sense or operate or relate to any of the phenomena out here, short of this little narrow band that we're sensitive to, and we simply had no idea that any of this existed. So what a good theory of electromagnetism allowed us to do were two things. First of all, unification. That is, we were able to understand that actually all of these phenomena, despite the fact that we've had for thousands of years different names for them, nevertheless are all one thing. In other words, and I'm going to lean on this throughout the talk, science is moving forward whether or not our ancient linguistic categories keep up with it or not. And I think they should keep up with it. But so what science told us is that actually those things are all the same, even though we have different words for them. And two, the fact that because we have this much better theory of electromagnetism, we can now make technology that allows us to operate along regions that were previously inaccessible to us, which improves the quality of life. So what I'm going to argue is that the exact same situation currently exists in the field of diverse intelligence or cognition more broadly. That basically we have lots of phenomena that people, because of these ancient linguistic categories, people insist on are different. People talk about crisp categories of things like living organisms and machines, and they like binary kinds of labels, as in this is intelligent, this is just physics, and so on. And I'm going to argue that we're basically in the same situation here, where we can now develop a good conceptual frameworks and the technology that they enable to move beyond these limitations. And so one of the things that I'm working on is a framework that enables us to recognize, create, and ethically relate to very diverse agents. So this includes, of course, the familiar animals and maybe some more unusual things, whales, octopus, people like to think about those things, but also some very unusual creatures like colonial organisms and swarms, engineered new life forms, so synthetic morphology, AIs, whether software or robotically embodied, perhaps someday exobiological agents, and then actually some even much more unusual kinds of things, which we probably won't get to today, but I can talk about them if people want. In the Q&A, which are not even strictly speaking recognized today as physical beings at all, and these are patterns within media. So my goal is to develop a framework that allows us to think about all these things and understand what they have in common. And very critically, it has to move experimental work forward. In other words, not only should it drive new discoveries in biomedicine, in bioengineering, but it also should drive improvements in ethics, in our ways to relate to other beings. And the first version of it is here in this very lengthy paper that tries to lay this out in great detail. Of course, I'm not the first person to try for something like this. Here's Rosenbluth, Wiener, and Bigelow. and around 1943, talking about the cybernetic perspective of how you get from passive matter all the way up to human level metacognition and things like this. But if you want, you can sort of emphasize these transitions, or you can think about it as a spectrum. But either way, I think that what we're dealing with here is what I call the axis of persuadability. In other words, I take a very for the purposes of this discussion. I take a very engineering approach to these things, where I think agential claims are basically interaction protocol claims. That is, when we say something has a degree of intelligence or cognition or agency or whatever, we are not really trying to debate the essence of what it truly is, some sort of ground truth that we can all agree upon after we fight about it for a while. Actually, what we're saying instead is a very observer relative claim that I'm going to use a set of tools, maybe hardware rewiring, maybe control theory and cybernetics, maybe behavior science and rewards and punishments and things like that, maybe psychoanalysis and love and friendship and whatever else happens at this end of the spectrum. So I have a set of tools, and I'm going to use those set of tools to interact with the system. And then we can all see how well that worked out for both of us, meaning that it is a publicly observable empirical claim that a certain set of tools is appropriate to a certain system. And so this is very important, because it means that we can't sit back in our armchairs and philosophically have feelings about this. I don't think we can say, well, this is just a chemical reaction. It definitely is not conscious or cognitive or whatever. Nor can we say, wow, there's a spirit under every rock. The whole universe is this beautiful mind together. I mean, maybe, but you can't decide that just sort of, based on feelings. You have to do experiments, and you have to show which of the kinds of tools that we're used to, or in fact, new tech tools that we have developed, give you any kind of a purchase on the system. So that's what I'm interested in. I will also point out, though, that in moving up, and sometimes I give a talk that sort of goes across all of these increasingly more unusual kinds of agents. In moving up, what happens is sort of what happened when we discovered new kinds of numbers. We had counting numbers, and then somebody came up with zero, and then the negative numbers and fractions, and then irrationals and so on. And the interesting thing is that in widening your view of what a number is, going from this pre-scientific counting thing to some very weird, transcendentals and so on, there's a few things that happen. You have to break conceptual categories, right? You have to break assumptions that you've had, and actually, this is quite painful. Some people, like this poor guy, Hippasus, he was thrown off a ship apparently for breaking some assumptions about rational numbers and things like that. And so I think what we have in the field today is a kind of mind-blindness, where we are stuck on some ancient categories that prevent us from seeing and relating to an important part of the world. And getting past that certainly does make things more complicated. And it upsets a number of established categories. People get really upset over this stuff. I know, because I get the emails from everybody who's kind of ticked off in either direction based on the things I'm going to say. But I really do think it makes for more meaning in the world, and it makes our experience more beautiful. So specifically, what I think we need to do is to get beyond this kind of ancient vision of our relationship to the world. This piece of art is called "Adam Names the Animals in the Garden of Eden," and of course, what it's telling us, and I think this has to be discarded, is this idea that there are discrete natural kinds. We all know what these animals are, and then here's Adam, and he's separate, he's different from all of them. So all of that, I think, has to go. But the good part of this, and the very deep, I think, part of this piece of art, is that in this ancient story, it was up to Adam to name the animals. God couldn't do it. The angels couldn't do it. Adam had to be the one to name them. And that's for two reasons. One is because in those ancient traditions, naming something meant you've discovered its true inner nature. And this, I think, is very important, because we are going to have to, as a species, we are going to have to discover the true inner nature of all kinds of weird creatures that I'll talk about, and give them names in that sense, that we must begin to understand them. And of course, the other thing is that it was up to Adam to do this, because he was the one that would have to live with them. and all of us are going to be living in a very different world moving forward, I think, and so this has to be updated. Now let's just talk about some different kinds of agency. First of all, it's very easy to detect this kind of thing in brainy mammals. So here's this creature has a very good theory of mind. It sets up this little accident scene right there, puts it right on the neck there, and because he knows what reaction he's going to get out of his human owners, and in fact, if nothing happens too quick, he's going to look. Are they watching? Yeah. So it's very easy when we see things like this. We know exactly what's going on. No problem, and that's because they operate at the same scale of time, of space, of direction. They have the same, very similar goals to our basic fundamental goals, and they operate in the same space, the three-dimensional space. Although I will point out that even that is not so simple. And there's this paper that Karina Kaufman and I wrote recently, reviewing some cases of humans with very minimal brain structure, but normal or above normal cognitive performance. So actually, even with brains, things are not simple, because the mapping of the amount of brain real estate you have to the level of performance you're able to muster is actually not straightforward at all. I mean, these cases are rare, but they exist, and so they're telling us that we actually don't understand even this relationship. We don't understand very well. But let's move beyond that. The thing is that all of us, whether we're talking about our bodies or our minds, are a kind of self-organized pattern in a substrate. So this is an early embryonic blastomere here, and we look at this blastoderm rather, and we look at this and we say this is one embryo. What do we mean? What are we counting when we say there's one embryo? I mean, there's hundreds of thousands of cells here. What is there one of? Well, what there's one of is commitment. There's one story that all of these cells have bought into in terms of what they are going to do in anatomical space. It's a very convincing model of themselves and later of the outside world that binds them together into a single vision, which we then have named an embryo. And in fact, if you put little slices into this blastoderm, and I've done this in duck embryos when I was a grad student, what will happen is that each one of these regions will not be able to sense the others. It will self-organize into an embryo, and you will end up with twins, triplets, and so on. So the number of individuals, the number of agents that can emerge at this higher level out of this continuum is not fixed. It could be anywhere from 0 to probably half a dozen or more. It's not set by the genetics. It's a dynamic, active process having to do with alignment, both physical and the cognitive alignment between the competent subunits. And this is true for our bodies, but it's also true for our brains and our minds, because in terms of split brain patients and dissociative identity disorders and so on, there are potentially multiple human personalities or human agents within one brain. So again, this relationship between the actual hardware and the real estate versus the agents that inhabit it is kind of not simple at all. And so now we can move to actually talking about something even less well understood, which is biological intelligence outside the brain. And we're gonna need to realize that intelligence is way older evolutionarily than brains. This idea breaks our common focus on the specific scales and substrates of intelligence, and we're going to have to be able to recognize these things using some new markers and some expansion of our imagination, and it has many useful applications. And what I'm going to do is tell you how to apply this kind of thing to one particular model system, which is cells, so living cells, as a collective intelligence operating in anatomical space. Okay, that's this section. So let me unpack that for you. This is the sort of thing we're made of. Now, this happens to be a free-living organism called a lacrimaria, but you get the idea. Single cells, no brain, no nervous system, extremely competent in its tiny little agendas that it has, and this is the kind of thing that we're made with. It's an agential material, very different engineering with this material than you would with passive matter like wood and metal. In fact, even below that level, so never mind the cell itself, but it turns out that the molecular pathways inside of cells, so signaling pathways, gene regulatory networks, that kind of thing, are capable of several different kinds of learning, including Pavlovian conditioning, sensitization, habituation, Pavlovian conditioning, so association. They can count to small numbers, some amazing properties just of the molecular networks. Never mind cells or synapses or brains or any of that. And in our lab, we're taking advantage of this to build devices to actually train the molecular networks inside of cells to be able to do useful things like drug conditioning and so on. So the thing about our bodies is that it has nested intelligence. That is, we are made at every scale. We are made of a material that has agendas. It has problem-solving competencies. It has the ability to navigate various spaces. We call this the multi-scale competency architecture. So all the way from the molecular networks up through the different scales up to swarms, these things navigate different spaces. And the thing is that because of our own evolutionary history, we are very obsessed with three-dimensional space. So movement in three-dimensional space is to us a fundamental feature of embodiment. In particular, we can only recognize easily medium-sized objects moving at medium speeds through three-dimensional space, like these kinds of things. Anything else really requires a lot of imagination and a lot of expansion of our categories. For example, if you think about artificial intelligence or even brain organoids, people will often tell you they're not embodied. Okay, their intelligence is not real, so to speak, because they don't have a body. They're sitting there inside some server box somewhere. The problem is that we are actually very bad at recognizing embodiment. If it's not rolling around the floor in your lab or in the environment, we don't think it has a body. But biology shows us that actually this loop that underlies intelligence, meaning sensing, decision making, memory, and then actions. That loop, so prediction and those kinds of things are operating in many different spaces besides three-dimensional space. So cells and tissues navigate the space of gene expression. They navigate the space of physiological states. They navigate anatomical morphospace, which is what we're going to be talking about soon. And all of these things are not trivial for us. In the same way that mathematicians have learned to think about high-dimensional kinds of shapes and patterns and so on, which we can't really visualize, we're going to have to do the same thing here. We're going to have to develop tools, technologies basically, that help us understand how intelligence navigates these spaces. So when I say intelligence, what I mean is William James's definition: some degree of the ability to reach the same goal by different means. Okay, I love this definition. It doesn't say anything about brains. It doesn't say anything about the origin story of the agent, how whether they were engineered or evolved. It doesn't say what space the goal is in. It just says that it's the ability to navigate that space to reach the goal when things happen. So the hypothesis that we've been following that I'm going to tell you about today is that morphogenesis, meaning embryonic development, regeneration, metamorphosis, cancer suppression, all of those things, is basically the behavior of a collective intelligence exerting its competencies in anatomical space. And so now I have to cash this out with actual data. I have to show you what kind of collective intelligence do these swarms deploy? What problems do they solve? How do they navigate the space, right? And so it's actually very, very similar to what happens in the brain, except that it happens at a different time scale. It happens in a different space. But the mechanisms are actually all the same. And actually all of the tools of cognitive and behavioral science all work as well. And we've made a tool. I used to do this with my students by hand, and now we have a tool to do this, and you can play with this, where you can take any neuroscience paper and basically do a find replace. Anytime it says neuron, just say cell. Anytime it says millisecond, say minutes or hours, and you've got yourself a developmental biology paper. And we have a system now that will do that for you. So because there is a fundamental symmetry between the self-assembly of the mind and the self-assembly of the body. So let me show you some interesting things that agents that navigate morphospace that they can do. First thing they can do is they can get to the same goal from different starting positions. So this is an axolotl, highly regenerative amphibian. If you amputate the leg at any position, no matter where you do it, the cells will grow very rapidly. They know immediately that they've been deviated from this target state. They will rebuild the limb, no matter where they started from. They will rebuild the correct amount of stuff, and then they stop. And that's the most amazing thing about regeneration, is that it knows when to stop. When does it stop? It stops when the goal has been reached, when the delta, the error, because this is basically an example of homeostasis. It tries to reduce the error between where it needs to be and wherever it is now, and it can navigate in anatomical space to get back to where it needs to be. By the way, embryonic development is really just an extreme form of regeneration. you're now called upon to regenerate from just one cell. This thing regenerates from a little stump. In embryogenesis, you regenerate from a single cell. And again, it can reach that from different starting positions, because if you cut early embryos into pieces, you don't get half bodies, you get perfectly normal twins and triplets and so on. So again, navigation of anatomical space from different starting positions. But it's interesting, it's not just about damage, it's about top-down holistic control. So here's another example in amphibians. If one takes the tail of one of these creatures and surgically grafts it to the flank back here, what will happen is that over time that tail becomes a limb, and the little cells at the tip of the tail, which are locally fine, they're tail tip cells sitting at the end of a tail, they become fingers. Okay, so what's happening here is the same thing that happens in any significant cognitive system, where the low-level molecular mechanisms are being harnessed, they're being controlled by a higher-level goal. The fact that this system knows, just like it knew what the goal state of a proper limb is, so that it can regenerate, it knows that at the level of the whole body, you don't have a tail in the middle, you have limbs, and so that delta, the delta between the memory of what it needs to do in the current state, is actually what drives all the molecular processes downstream that implement that state. And if this sounds weird to you, think about what voluntary motion is. So you wake up in the morning, you have very abstract high-level goals, financial goals, social goals, all this very abstract stuff. But in order for you to get up and walk and execute on those goals, those high-level abstract ideas have to be transduced into the movement of potassium, sodium, and calcium ions across your muscle membranes. So the biochemistry is actually controlled top-down by these extremely abstract strategies for navigating some kind of space. And basically, the way that this happens is through, and I could show you, by the way, I've skipped already a lot here because for reasons of time, we could do hours just on these various kinds of examples, because some of them are truly amazing. But I want to point out that the way that this navigation is guided is actually very similar to what happens in the brain. So in the brain, which is the animal brain is a kind of a non-controversial example of an intelligent system that pursues goals in some kind of problem space and so on. So the hardware is a bunch of cells that have these ion channels in their membrane, so they can adopt a certain voltage state. Then they have these controllable electrical synapses called gap junctions, through which they can propagate that electrical state to their neighbors. And so the electrophysiology of the signals through that network is what's thought to underlie the cognitive content of that mind. And for this reason, there's this project of neural decoding, where people try to read out the electrical activity of the brain and try to decode it to know what that the human or animal is thinking. So you should be able to decode the memories, the preferences, the goals, all of it should be decodable. And what happens here is that this electrical system controls your muscles, so it issues commands to your muscles to move you through three-dimensional space towards meeting your adaptive goals. Well, what an amazing system. Turns out it's very old. Long before brains were here, evolution discovered this kind of hardware-software feature of electrical networks, and all cells in your body have ion channels. Most of them have gap junctions to their neighbors, and long before there were any brains or neurons, these electrical networks were processing information to control your cells, not just your muscles, but all your cells, to move your configuration through anatomical space. And so now we as we have been for now over 2 decades, can begin on this parallel journey of asking, we know what the electrical networks of the brain think about. What do the electrical networks of the body think about? What were they doing before there were any brains? And could we decode and extract the cognitive content of those pre-neural bioelectric networks? So again, the reason I'm focusing on all of this is that it's not that bioelectricity is kind of a unique home for cognition. It's just an incredibly convenient cognitive glue. Evolution loves it, and because it's a very convenient way to allow competent subunits, such as neurons in your brain or cells in the rest of your body, to become a network that can process and synthesize information in spaces that none of the individual components could. So no cell in that salamander limb knows what a finger is or how many fingers you're supposed to have. but the collective absolutely does. And this is exactly why the bioelectric cognitive glue is exactly why you know things that your neurons don't know, and you have goals and spaces that your neurons couldn't possibly have, and why morphogenesis is able to do things that no individual cell knows. So in order to do this, we developed some tools. We developed the first molecular tools to read and write electrical information into these networks. So here you can see voltage-sensitive fluorescent dyes. These aren't simulations. These are real. This is real data. So this is an early frog embryo where you can see all the electrical conversations that our cells are having. These are some explanted cells. The colors represent voltage, so the membrane voltage of the cell. So it's like it's basically like reading electrical activity in the brain, except we can do this in other tissues. And then we have lots of simulation and computational analysis tools by which, at the level of the transcriptional ion channel network, at the level of the tissue, voltage patterns, and actually at the level of the whole animal, such as with this pattern completion kind of thing, we can analyze the dynamics of and try to understand what guides the changes in these patterns. Just to show you a couple of quick examples. This is something we call the electric face. So again, the different grayscale values are voltage here, and you can see this is a time lapse of an early frog embryo putting its face together, and this is one frame out of that video. You can see here that we can actually see what long before the genes turn on to regionalize the face. You can actually see here's where it's going to put the eye, here's where it's going to put the mouth. The placodes are out here. This is the readout of a pre-pattern that shows that this tissue, this anterior neuroectoderm tissue, already has this pattern laid out in terms of where the organs are going to go. And this pattern is critical. If you change it, the face will change. And I'll show you that in a minute. Not only does bioelectricity serve as a cognitive glue binding cells into multicellular assemblies, it also works on a higher scale between individuals. So here are some Xenobots, which I'll talk about later. When we poke one, within seconds, these other two find out about it, and the same thing is here with embryos. You know, this calcium wave that you're seeing here shows, and we've done studies showing that the collective is actually able to do things that no individual embryo can do. We can talk about that if anybody has questions. So these kind of dynamics serve as multi-scale cognitive clues. Now, more important than just tracking these patterns is being able to rewrite them. So you want to be able to put information in, not just detect it. And so we've developed tools for that, basically all stolen from neuroscience, because it's the same set of mechanisms. And this is what's very important. If the tools don't distinguish between neural bioelectricity and the things that go on in brains and the kinds of things that go on in other tissues, maybe are harsh distinctions between university departments and journals and funding bodies that say neuroscience is this and it's cognition, and then this is just developmental biology. It's not the same thing. Maybe those things are not as disparate as we try to make them. And to do this, we don't use any applied fields. There are no electrodes, no magnets, no frequencies, no waves. We don't do any of that. What we do is manipulate either the gap junctions, so control the electrical topology of the tissue network, or directly control the ion channels, meaning using optogenetics, using drugs, and so on. You can open and close channels, which changes the voltage in predictable ways. So this is the communication interface that we're hacking. All of the stuff I'm about to show you only works. It's not because we're so smart. It only works because this is how the cells usually control each other's behavior. This is how cells control other cells in networks and make sure that their activity serves a higher level set of goals, such as anatomical goals, right? And so that's what we're hacking here. So the first thing I just want to show you is an example of what happens when a very convincing bioelectric message is propagated from one agent to another. So these two are adjacent frames of a video that I'm going to play in a second, and what you can see, again, the color is voltage. And what you can see is that this cell that starts out a very different color, as soon as it touches this whole mass here, boom, it changes voltage. All it takes is one little touch. So here, I'm going to play this. So here it is. It's moving along, and mining and so business. This thing reaches out, bang, touches it, and right there you can see now it's got a different voltage, and now it comes and it joins this collective. And it's a whole story on cancer and voltage and why cells do or do not play nicely with other cells that we can talk about. But I want to show you the example of an informational takeover. What's happening here is that once this cell touches it, that bioelectrical state is taken up, and even after it's left, that's it. Its physiological state has changed, and all of the things it's going to do later have changed. And I'm showing you this because this is the kind of thing we really are interested in being able to duplicate for biomedical reasons. So for example, suppose that you wanted to convince a bunch of cells to grow an eye, as you might want in a medical application. Well, it turns out that you don't actually need to know all the steps that are required to build an eye, or how to tell which genes to turn on and off, or how to put the stem cells where they go. Eyes are very complex. We have no idea how to build an eye. But what we do know is how to provide a very top-level prompt, and I'm using these words on purpose, a high-level prompt that basically just says, build an eye here. And the way we do that in this, for example, in this early frog embryo, we introduce some RNA encoding a particular potassium channel that is going to establish that little voltage spot in a group of cells, just like what you saw in that electric face. And when you do that in a bunch of cells that normally would become gut, so here's a tadpole side view, here's the mouth, the brain up here, the eyes, the gut, and here's the ectopic eye. And if you look inside, it has all the same lens, retina, optic nerve, has all the same stuff inside. And so the first thing you learn is that these patterns of bioelectric state are instructive. They actually determine what happens next. So they're information patterns within this excitable medium of cells that actually matter to what happens next. They're extremely modular. We didn't talk about which cells are next to which other cells. We just gave an organ level instruction, make an eye. They did all the rest. Just like in that top-down control that I was talking about before, how your high-level goals can make the biochemistry of your body respond appropriately. That's exactly what's happening here. We're letting it do all the hard work. In fact, it's amazing. It has amazing competencies because if you only inject a few cells, so these blue cells are the ones we injected, all this other stuff out here that make this nice eye, we didn't touch any of it. It's the blue cells that are telling the other cells, hey, you should help us build an eye. There's not enough of us. You need to participate in this process. The amazing thing is that what's happening here is really a battle of models of the future, or worldviews, if you like, for cognitive systems, because the cells that we inject here are saying to the other cells, you should be an eye. And at the early stages, you might see in embryos, you might see 1, 2, 3, 4, 5. You might see like all kinds of ectopic eyes. But then towards the end, some of them disappear and all you get is one. What happened to them? Well, it turns out what happened to them is that there's a cancer suppression mechanism that resists this message. So we put in, so these cells are saying to the neighbors, you should really be an eye. The other cells, which have an evolutionarily built-in process that says if your neighbors have some sort of weird voltage, you need to equalize them out. You need to normalize them. So the others are saying no, you should be skin or gut or whatever it is that we were. And sometimes the ectopic eye wins, and sometimes the skin wins. And we can see this bioelectric pattern trying to take over. Become convincing. Change the set point, the actual set point of the cell, so that they can build to those structures. So we can see this information propagating and competing for the attention of the medium. And for biomedical purposes, we would love to be very good at this. You want the cells taking on whatever message you're giving them, not fighting you on it, which is actually what happens to many therapeutics today. So one of the interesting things about all of this is that the border between the agent and its world is quite plastic. I call this border the cognitive light cone. It's not the size of your sensory range or your effectors or anything like that. It's the size of the biggest goal that you can maintain and pursue. So individual cells have little tiny cognitive light cones. They're mainly only interested in things like this, homeostatic loops about pH and hunger level and things like that. They have a little bit of memory going backwards, a little bit of predictive capacity of the future, but it's all in a very small spatiotemporal scale. But when cells assemble into groups, they start to take on grandiose goals. I mean, these cells here have this giant thing as a goal, this limb. Again, no individual cell knows what this is, but the collective now, because they are connected into a bioelectric network that scales the agency of the components. It first of all enlarges the intelligence. The cognitive light cone gets bigger. It can solve much bigger, more interesting problems. But also there's a sense of a loss of agency for the subunits, because what the larger level is doing is bending the option space so that everything they do is in service of this larger goal. So the agency is actually moving as the cognitive light cone and the border between self and world shifts. And this strange way of thinking about things has very practical implications. For example, in cancer, and this is what happens when we inject human oncogenes into this tadpole, there'll be a tumor. You can already see the cells electrically decoupling from their neighbors, and what they're going to do is they're basically going to treat the rest of the body as external environment. Once they're decoupled, they're just amoebas now. Their cognitive light cone shrinks, and they're not more selfish than normal cells. They just have smaller selves. In other words, the boundary of between self and world, instead of working on this nice organ, is now shrunk to the level of a single cell, like their unicellular ancestors. And so that story of cancer has very practical implications. We said, okay, what if we don't kill those cells, we don't fix the genetic defect in this oncogene, all we do is forcibly reconnect the cells to their neighbors. Okay? And when you do that, by injecting certain ion channels, this is the same animal. Here's the oncoprotein blazingly expressed here, in fact, all over the place. If you were to sequence it, you would make a prediction that there would be a tumor. Of course, you'd be wrong, because there is no tumor. Because it's not the genetics that drives any of this. It's the physiology. It's the computations and the information processing and the cognition within the tissue that tries to set goal states. So these cells, even though they have this nasty oncogene, they're working on making nice skin and nice muscle because they're not individual cells. They're part of a cognitive collective. So all of this, and there's many other things I didn't get a chance to show you, is really part of a new approach to biomedical interventions. So the current approaches are very bottom-up, very hardware-focused, as computer science was in the 40s and 50s. To program a computer, you had to physically rewire the hardware. But now we have the ability to talk about all sorts of other approaches that exploit the intelligence of the material. Okay, and I think future biomedicine is going to look a lot more like somatic psychiatry than it's going to look like chemistry. It is not going to be about the molecular hardware and if you're interested in that kind of stuff, you can see all of that in these papers. But the reason I bring this up, this medical stuff, we could talk about this for hours. The reason I bring it up now is just to point out that all these deep philosophical questions about what is intelligence? What's a good way to rigorously quantify cognitive capacities? How do agents set the boundaries between themselves and their world? These deep ideas are moving from philosophy to empirical science. By having these kinds of frameworks that we've developed, we've actually reached all sorts of novel discoveries that are moving towards therapeutics. There are companies, and I'll do the disclosures at the end, that are now licensing all this stuff for therapeutic approaches because it allows you to find new things. And I think, in fact, very practical things. And I think that's a key question as we start to debate different ways of looking at cognition. We need to keep in mind the kind of empirical outcomes of different worldviews. Every model helps you see certain things and it hides certain other things. And by these kind of outputs is how we can judge them. So in the second half of the talk, I want to move beyond that kind of a thing and get a little more weird in terms of what other kinds of agents there are. And we're going to talk about non-biologicals, some hybrids, and even some really strange stuff, minimal agents. And this is going to break some other categories. For example, this standard debate that people often talk about, whether it's a life or a machine. I think these binary categories absolutely have to go. And one of the reasons they have to go is because it isn't just about AIs or humans or any of that, but we have to realize that this modern adult human that is kind of the center point of a lot of work on the philosophy of mind is not a binary category. It's at the center of a continuum on a developmental scale and evolutionary scale, starting with one cell. So if you think this human has some sort of special properties, it has moral worth and responsibilities and choice and all these things. When exactly did that show up? Because there is no magic lightning flash in development where you go from physics and chemistry to being a mind. It's a slow, gradual scale up. And the same thing during evolution. You have to ask yourself, which of our hominid ancestors would have these properties? And so that's one continuum. And that reminds us that, knowing what we know about developmental biology and evolution reminds us that a continuum hypothesis should be the null hypothesis. People say all the time, there are humans have real cognitions, cells don't, and if you think it's a continuum, you have to show why. I actually think it's the exact opposite. The facts of developmental biology and evolution tell us that the continuum hypothesis is the null hypothesis. If you think there are sharp categories, you have to say where does the magic come from and why, and things like emergence and so on. We'll talk about that. But there's another continuum. which is the continuum of technological and biological change. We know, and already there are modified humans in various ways walking around, and there will be much more. And so, if you want to keep these kinds of distinctions, you're going to have to know what to say about this kind of thing, which is going to be very prevalent. You're going to have people with different engineered components, and I don't think any of us want to be trying to measure and see whether this guy is 51% machine and so on. The idea is that biology is so interoperable that almost any combination at any level of evolved and engineered material is some kind of agent. And so now we have to ask the following fascinating question. We know what the for example, behavioral and morphogenetic goals are of standard evolved beings, because evolution presumably set them. That's what people generally think. So now we ask, these kinds of beings with no evolutionary history behind them, what are their goals going to be? What are their properties going to be? What are the cognitive capacities of these very different kinds of beings? And so if we can't rely on genetics and we can't rely on evolutionary history, what else is there? Right, and we study goals in morphospace a lot, and different different head shapes in planaria, and how the same hardware can visit different anatomical attractors, but these attractors were set by evolution. If you don't have that to rely on, what else is there? And I think we need to start thinking broadly. First of all, we need to understand that there are patterns, very complex, strictly defined patterns that come from neither genetics nor environment. What I'm showing you here is what's called a Halley plot. This is just one example. I like these things because they kind of look organic and they're kind of pretty, but there's a million versions of this. It's a Halley plot of a very simple mathematical function in complex numbers. That's it. This is the complex function, and they make all sorts of and this is a video basically made by changing these parameters slowly in each frame. So you're looking at all this stuff, and you can ask yourself, okay, what exactly determines the precise shape of this? If you're a biologist, you would say, well, it's going to be either physics or it's going to be a history of selection. That's what we have. But these mathematical kinds of structures have neither. They have neither a history of selection, nor are they dependent on any facts of physics. They come from a different world, and we're going to talk about that a little bit. And so now we can say, okay, if there are patterns that don't require either a genetics, any kind of a genetic history, nor do they depend on laws of physics, how relevant is this to biology? And I just want to say two things. First of all, questions in biology often, I think, exit the world of physics entirely. So there are cicadas, for example, that come out at 13 and 17 years. Why? Because they're trying to time it so that it's harder for predators to latch onto those cycles. And if you want to understand why that works, the answer is because 13 and 17 are prime. That's why. And the answer to why are those numbers prime is nowhere in physics, and it's nowhere in biology or evolution. Okay, you've left the set of physical explanations, but it's crucial for you to understand the biology that's happening. So let's look at some biological forms that appear to make use of some of these kinds of things that are not from direct history. First of all, these are Xenobots, and Xenobots arise when we take some cells from the animal cap epithelium of a frog embryo. We liberate them from the rest of the cells, and what they do is they pull themselves together into this little kind of construct. I want to show you what some of the cells do. Each one of these circles here is a separate cell, and there's a group of them here. I think it's great fun that it looks like a little horse that sort of moves as a unit. It's over here kind of examining these other cells. We actually don't know what any of the signaling is yet, but you'll see a little calcium flash here. So there's all kinds of rich behavior. But when they get together, first of all, they use the cilia on their surface to swim through the environment. They can go in circles. They can patrol back and forth. We can make crazy kinds of shapes like this thing. They can have collective behavior here. There's groups of them. This is what it's like for a Xenobot to traverse a maze. So still water, no water movement. So it makes the corner without having to bump into the opposite wall. So it takes that corner. Then it spontaneously turns around, goes back where it came from. Okay, these are not like conventional biobots that are made of muscle that you have to pace with electrical stimulation. This is completely autonomous. This thing does its own behaviors. Here's one amazing behavior that it does. We call this kinematic self-replication. So if you provide it with loose epithelial cells, what they will do, both collectively and as individuals, is run around, make little piles of the material, and because it's dealing with an agential material, just as we were when we made the Xenobots, that little pile matures into the next generation of Xenobots. And guess what they do? They do the same thing, and they make the next generation and the next generation. So the idea here is that this is like von Neumann's dream. It's a robot that's able to make copies of itself with material it finds in the environment. So that's pretty wild. Now, one of the things that that you will see in these bots is this kind of calcium flashing, and this is the exact same technique that are used to characterize cognitive activity in brains. In other words, we can take these electrophysiological signals and apply some tools to them. And this, by the way, is the work of Thomas Varley from the Bongard lab. And when we apply these kinds of tools, you can, for example, look at something called phi-R, which is a measure of integrated information based on Phi 2.0 by Balduzzi and Giulio Tononi. And what we see is that compared to null models, there is significant integrated information in these structures, just like there is in brains analyzed with fMRI. And my point is not that these things are like brains. My point is that both brains and these structures, which have no neurons at all, have an interesting information property that basically quantifies the extent to which something is more than the sum of its parts. That's the point of this kind of causal information theory. So we can start studying, again, this is in the name of stealing all the tools from neuroscience, mainly because I don't believe that neuroscience is about neurons at all. I think neuroscience is a study of cognitive glue. It just so happened that we latched onto the brainy kind of examples first. But what it's the study of a scaling of agency from competent subunits to collectives that express that competency at larger scales towards larger goals in other spaces. So that's one creature I wanted to show you. Another one is this one that. If you saw this initially, and I asked you, if I didn't tell you what this was, you might guess that this was something we got at the bottom of a lake somewhere. It looks like a primitive organism. You could guess at the genome and think that it would also look like a primitive organism genome, except that if you sequenced it, what you would see is full-on, totally normal Homo sapiens genome. These are anthrobots. They're made of adult human tracheal epithelial cells. They self-organize. They become this motile little thing that looks like no stage of early human development or of any human development. They have some interesting properties. Again, you can see some group behaviors here. First of all, these guys express 9,000 new genes differently than the original tissue. They have a completely altered transcriptome. Xenobots, by the way, express hundreds of genes differently. These guys express thousands of genes differently. About half the genome is now different. We didn't touch in either case, the Xenobot or the Anthrobot case, we didn't edit the genome. We didn't put in any synthetic biology circuits. There are no scaffolds. There are no nanomaterials. There's no weird drugs. All it is we liberate the cells. They have a new lifestyle. They reboot their multicellularity in a new way, and for that they drive a novel transcriptome. They can self-heal, as can Xenobots. If you make mechanical damage, they will repair themselves. And this is something kind of wild that I don't have time to talk about. They're younger than the cells they came from. This process of assembling yourself into a new being in a new lifestyle is actually rejuvenating. Kind of makes sense. We know our children are not born at the age of their parents. They're born at almost zero. So it's kind of the process of morphogenesis is drastically rejuvenating. But even these anthrobots are younger than the cells that they came from. They have a number of behaviors, and we can draw an ethogram of different types of behaviors and the probability transitions between them. And one of the cool behaviors that they have is that they can traverse these scratch wounds. So all this stuff up here are human neurons, and I made a big we made a big scratch through here with a scalpel that these bots can then sort of drive down. And what you'll see is that when they pick a spot to land, they assemble into this kind of superbot cluster. So there's, I don't know, maybe a dozen of them here. And what you see they start to do is they start to heal across the gap. If you lift up the bots 4 days later, you see that they've made this healing. So I want to point out two things here. First of all, this is a kind of intrinsic motivation in the sense that We didn't program them to do this. We didn't make them do this. You know, whoever thought that your tracheal cells, which sit there quietly in your airway and deal with you know, pollen and mucus and so on, have the ability to be a little autonomous being that can heal neural wounds. We would have no idea. We didn't make them do this. That's the first thing, and it's kind of cool that the one we found is actually very positive and healing, right? I like it. And then the other thing is to point out this is just the first thing we looked at. This is not you know, test 92 of 1,000 things we looked for. This is the first thing we looked for and that we found. So that says to me this is probably just the tip of the iceberg. There's probably a million things that these guys know how to do. So here's the important point here. Where do the properties of all these kinds of beings come from? We know that the frog genome, for example, gives us this. Okay, so the specific history of the frog genome, by butting up against the environment, gave us a certain set of developmental stages and then these tadpoles with certain behaviors. But it also gave us this, the Xenobots. This is an 84-day-old Xenobot. So what did the genome actually learn during all this time? There's never been any Xenobots or Anthrobots. There's never been selection to be a good Xenobot or a good Anthrobot. You can't really blame the properties, the forms and the behaviors of any of these kinds of beings on a history of selection. They were never selected for this. Now, what you could say is that, well, the processes of evolving a frog automatically sort of give you this stuff. And that's fine, except that what that does is break the whole point of what evolutionary theory was supposed to do for us, which is to explain to explain the specific properties that living organisms have with respect to the history that got them there. There should be incredible amounts of specificity between what happened in the past and what we observe now. That's kind of the whole point. And if we're saying that, well, at the same time that you selected for all this, actually you got all these capacities, then what that's telling us is that the standard story is incomplete in major ways. And by the way, on the computational side, you can ask the same question. We know when the computational cost for all of this was paid. When was the computational cost for all of this paid? Because this never happened before. So we have some very interesting questions around when these kinds of things appear and why do they appear. And again, I'll just point out that I think it's very clear now that the thinking that the physical world is closed with respect to causation and with respect to the things you need to understand to explain physics, biology, and computation is just no longer viable to think that it's closed. It hasn't been, I think, since the time of Pythagoras and Plato. They saw this very, very clearly. And you know, this, I already mentioned this idea of these you know, the properties of prime numbers are actually what you need to understand if you want to know why the cicadas are doing what they're doing. This is not a fact of physics or evolution. This is a fact that comes from mathematics. So now here comes you know, some ideas about how to think about this in the future. And I realize that this is a step that most people do not want to take because they like a nice sparse ontology where the physical world is all there is. I don't think that's viable. So how do we think about this? There's a couple of ways to think about these competencies of novel beings. One way to think about them is the fact is that evolution exploits a ton of these free lunches from mathematics, properties of networks, of numbers, of computation. Evolution uses them a lot, and we could go through some examples. So how do we think about them? So here's the typical way people think about them. They say, look, these are just facts that hold. You know, you found that Xenobots do something that they weren't selected for. Meh, it's just an emergent fact that holds in our world. That's it. And so the problem with this standard view of emergence and complexity is that what that basically, well, the benefit is that you keep your sparse ontology. You can just say that all we have is the physical world. But the problem is it's a very Mysterian and I find very pessimistic view of science. It just means that, you know, emergence, meaning anything that is surprising to us, anything that we didn't see coming, is, you know, you can write it down in your big book of emergent amazing things, and then what? You know, thinking about this as a disordered set of surprises, I just, you know, metaphysically, I don't like it at all. I prefer what mathematicians do. So Platonist mathematicians believe that there is an ordered latent space, a non-physical ordered space of patterns, such as the truths of number theory and all this kind of stuff, which can be studied systematically. Okay, I find this a much more optimistic agenda because it's not just a bunch of surprises. If you think that these things exist in an ordered, structured space, you can then systematically traverse that space and discover those patterns. And so I think that all of these things that we build, Xenobots, Anthrobots, all this stuff, but also normal embryos and cells and robots and everything else are basically interfaces to this Platonic space of patterns. They are vehicles that we can use experimentally to map out the space and to understand the mapping between the interfaces that we build. I think physical bodies are interfaces to these patterns, and we need to map out the relationship between these pointers or interfaces and the thing itself. And so again, just to say that there is a precedent for this. Large numbers of mathematicians think that is what they're doing when they are doing their science. Of their study of mathematics, but I will go one step further and simply hypothesize this, that along with the layer of the Platonic space that contains low agency forms, such as facts about numbers and shapes and so on, are very high agency forms, very complex, dynamic, high agency forms that we would recognize as kinds of minds. They are basically forms of behavioral propensity. They are behavioral properties, not only morphogenetic properties as you find in seashells and other things that obviously are implementing some mathematical forms. So that's what I think, that all of these things that we're building here or that we find in the natural world are basically interfaces or pointers into this space that we now have to understand. So I'm gonna end in a couple minutes and just finishing up this way. That first of all, I think that Garden of Eden kind of view that we started out with, I think is going to be way weirder than anybody imagined. Right? I think we really do need to start thinking about minds that are in very, very different kinds of embodiments, and overall, this idea, what I've been pushing now with this concept of facts of biology that are not explained by either physics or evolutionary history, means that we really have to think hard beyond the physical embodiment and asking, what is this interface actually pulling down from that latent space? But now, if that wasn't weird enough, I want to take one step further. If I haven't lost everybody yet, usually this is the point at which most people have sort of gotten off the ship that I was talking about. But here's the last bit. If that hadn't done it, probably this might. I want to point out the following, that it doesn't take much to be a good interface to interesting patterns from that space. In other words, I don't think it requires cells, life, or large complexity to have to be an interface to patterns, including patterns that we recognize as behavioral propensities or even problem-solving skills, aka intelligence. I don't think it requires any of that to start being the interface to ingressions to some of those patterns. And I don't have time. We're running out of time, but I could talk about it, and you can see this in this paper. Even something as simple as bubble sort. Okay, so a simple computer algorithm that was designed to sort numbers, and I specifically started this project because I wanted the most shocking example possible. I wanted something that was really simple, really deterministic, that had no more details to be discovered. It's like 6 lines of code. You can see everything there. There's nowhere to hide. And I wanted to show what even something as simplistic as that has. What we're finding is that not only does it have interesting problem-solving capacities, for example, delayed gratification. Okay, so it doesn't just follow a gradient; it's able to move away from its goal in order to recoup gains later. That's a pretty good skill actually for cognitive systems. Not only does it do that, it does these other weird side quests, which we call clustering, that are nowhere in the algorithm. They're doing specific things, literally that are absolutely nowhere in the algorithm, and so, much like us, they're forced to, by the physics of their world, to do certain things. So they're forced to sort numbers, and here you can see the sortedness rises over time. But they also do this other crazy thing called clustering, and they do as much of it as they can before the physics of their world grinds them down because they have to sort. It's a very kind of existential story of our life too. You know, we are also subject to the physics of our universe, but until we sort of disband as a collective intelligence, we have the opportunity to do some things that are neither demanded nor prohibited by the biochemistry that runs our life. And so that kind of life, that kind of feature of life, is actually appearing in a very simple, very basal form all the way at the beginning. And here we need to start to understand how we relate to engineered agents. Because, yeah, there are the things that we force them to do through reinforcement learning and things like that. but there's also the stuff that they do that we never ask them to do, like the things that I've shown you today of the cells that are going to build an eye, or the Xenobots with their kinematic self-replication, or the Anthrobots. They do things that we did not engineer them to do, that we did not ask them to do, that evolution did not prepare them to do. And this very interesting aspect of intrinsic motivation, I think, is a huge open research program for the future of agency. And so I just want to point out the big problem with all of this, and the reason a lot of people hate these kind of ideas, is that there's a whole community of organicist thinkers who really want to keep a sharp separation from quote-unquote machines and the magic and the majesty of living things. And they say to me, look, by putting these things on the same spectrum, you are devaluing what's amazing about life and mind. You are basically asking us to treat as machines things that we know are not well described by machine metaphors, meaning living and cognitive agents. And I just want to point out here that I'm going in the exact opposite direction. I am in no way saying that our simple machine metaphors are suitable for understanding life and mind. I'm saying the exact opposite. I'm saying that in our urge to preserve what's special about life, we have decided that there is this category of dead matter and these kind of machines that are not like that are basically, they only do what the algorithm or what the materials tells them to do. I think that is a fundamental error. And I think because we've mistaken our formal models for the thing itself. Magritte is telling us that our formal models of objects are not the objects themselves. And I asked our graphic designer to draw this kind of thing. It says in French, this says this is not a pipe, and then it says this is not a Turing machine. Yeah, there are no Turing machines. There's only our formal models of a Turing machine, and this idea that computationalism, this idea that our computer formalisms capture everything that simple machines are doing, I think is exactly as wrong as the idea that some people push that biochemistry and the laws of physics tell the entire story of the mind. Right? We are used to giving us a break on that one and saying that the rules of chemistry don't fully determine what you need to know about our minds. Right, and they also don't for these things that up until now have been assumed to perfectly match our formal models of simple machines. I just don't think that exists anywhere, and we really have to understand that our models are not catching everything that these things are doing, and we have to be a lot more humble about that. So the bottom line is, and I'm kind of over time here. I just want to say a couple of very very quick things. This is the new space that we're going to be living in. All of the natural creatures that Darwin was so impressed with, and when he said endless forms most beautiful, are like this tiny corner of the space here. Pretty much any combination of evolved material, design material, and software are going to feature ingressions of patterns that we are very poor at predicting. And so cyborgs and hybrids and chimeras of all kinds, we are going to have to find a way to live with them in a kind of ethical synthbiosis. And so for the last minute or so, I want to look at this. If we are trying to build a continuum, what is it that all actual beings have in common? I think this idea of a continuum and of scaling goes all the way down. But what do they actually have in common? And I just want to list those things right now, and then we can talk about it. The first thing they have in common is this flexible cognitive light cone. So all significant agents have some kind of a goal-directed cybernetic loop, and the size of those goals determine what kind of a being, and the space in which they play out determine what kind of being you are. So not only the kind of goals you can have, but actually this question of where do you end and the outside world begins, and we've talked about this. I don't have to go over this again. The next thing, which does bear some discussion, we could talk about that, is the need to interpret your own memories. Significant agents are time slices of a persistent self-telling story, during which at any given moment you don't have access to the past. You have access to the memory traces that past you left for you, so messages, right? So your memories are basically messages given to you by a past self, just like you can have messages laterally from other beings, and it's up to you to, in fact, you have to interpret those memories. You don't know what the physical engrams in your brain and body mean. You have to actively reconstruct a story of what you are and what the future is as an active agent. So all active agents, whether engineered or biological, are experts in confabulatory storytelling. They don't have allegiance to how these messages were interpreted in the past. They tell the best story they can looking forward. And the cool thing is that this is not only a fact about cognitive systems, it's the same thing about biology. The reason that we can have Anthrobots and Xenobots and all kinds of crazy things I didn't show you today, tadpoles with their eyes on their tails and whatever. The reason all that stuff works is because the genomic information is to be interpreted. It is not a determinant of what happens next. It is a set of prompts, and this is described in this paper. It's a set of prompts that the agential material of cells is going to interpret as best as it can. And if that turns out to be a frog, under normal circumstances, great. And if the circumstances are not normal, it will be a Xenobot or an Anthrobot, and it will pick things out of those genomic affordances that it needs in its new lifestyle. So I'm going to stop here and just summarize, because there was a lot of information. I'm going to summarize here. I think that we are in a good place now to develop and mature conceptual tools to detect other kinds of minds and unconventional substrates. We have all kinds of tools, and it's driving new technology to make new empirical discoveries, such as biomedicine and beyond. I think that there is a huge diversity in the kinds of intelligence and the spaces in which these agents work. We are terrible at noticing it. We have to work at this, and also we have a lot of hubris in thinking that we or even the complex life forms are special. I actually don't think it takes much to get onto that continuum. That spectrum goes all the way down. And here's what I think all significant minds have in common. They have responsibilities. They are responsible for setting their boundary. They're self-constructing boundaries. They have to interpret their own memories. So they're continually self-constructing stories of what they are and what that means in their environment and what they should do. They have the responsibility for hacking their own parts towards their large-scale goals. So you have to control your cells, which control the biochemistry and so on. And I think what life is, if we wanted to define it, and I think cognition is a superset of life, but if we wanted to define life, what I would say is that life is what we call systems that are really good at scaling their cognitive light cone and have been under pressure to optimize their adaptive storytelling. They are really great at interpreting the information that they get because they are constructed of an unreliable medium. The biological medium is fundamentally unreliable, which has huge implications for evolution, for cognitive science, and so on. And I think that physical embodiments are interfaces or pointers to a structured space of patterns of body and mind that can be systematically studied. That is the research program. So if anybody's interested in this stuff, there's lots more papers where we discuss these things. Most critically, I want to thank all the people who did this work. These are the postdocs and the students who did all this that are mentioned here. We have lots of amazing collaborators that have participated in all of this. Our funders who have supported this work over the last 30 years. And I have to do the disclosures. There are 3 companies that supported some of this work and have spun out of our lab. And most importantly, most of the gratitude goes to the model systems, biologically and not, that are teaching us about these things every day. So thank you.